plant development, and for further information, you read up on plant development separate in the individual Wikipedia article. So plant development is regulated by environmental cues or signals and the plant's own receptors, hormones, and genome. Moreover, they have several characteristics that allow them to obtain resources for growth and reproduction, such as meristems. See the pronunciation here.
Meristem.
Yeah, so meristems, which are meristems, also post-embryonic organ formation and differential growth. Yeah, so continue for those. So the meristem is a type of tissue found in plants. It consists of undifferentiated cells called meristematic cells, capable of cell division. Cells in the meristem can develop into all the other tissues and organs that occur in plants. These cells continue to divide until a time when they get differentiated and then lose the ability to divide. So here's the setup over here. This is the Tunica-Corpus model. Let's click it here.
Tunica-Corpus.
The Tunica-Corpus model of the apical meristem, or the growing tip there. The epidermal L1, see the outside coating, and the subepidermal L2 layers form the outer layers called the tunica, named after a tunic or coat, and the inner L3 layer is called the corpus. You have the Tunica-Corpus corpus model here of how this of the apical meristem. The inner L3 layer is called the corpus. So there's the corpus there, and then this is the coating there, tunica or the tunic. Cells in the L1 and L2 layers divide into a sideways fashion, which keeps these layers distinct, and whereas the L3 layer divides in a more random fashion. So yeah, so these ones divide sideways and etc. This one is more random. You can get them inside here, etc. It's all random spot. So these ones lateral growth, and this is a random growth there. Yeah, so circle again. So random growth in the L3, and then there's the lateral growth in the L1 and 2. Yeah, so random in L3, and then lateral in the other way. All right, so in biology, a tissue is a cellular organizational level between cells and a complete organ. Yeah, so between cells are small ones, a complete organ is the big one, and then in between that, you just call that tissue, and it's not an organ, it's not a complete organ, and it's also not individual cell. It's not an individual cell. Yeah, so it's organizational setup is in between them. So you have a bunch of cells, but they don't form a complete organ. Call that tissue. Very interesting. So development begins with a seed, which is an embryonic plant enclosed in a protective covering. Yeah, and let's go back here. So this part right here. So moreover, the plants they have several characteristics that allow them to obtain resources for growth and reproduction, such as meristems. So that's meristems, that sideways growth there. And then post-embryonic organ formation, after embryo, you form organs. You also have differential growth. It grows differently in different areas, etc. So let's go back here. So the development begins with a seed, which is an embryonic plant enclosed in a protective outer covering. Most seed plants are usually dormant, a condition in which the seed's normal activity is suspended. Seed dormancy may last weeks, months, years, and even centuries. And dormancy is broken once conditions are favorable for growth. And the seed will begin to sprout or grow, or grows. It begins to sprout or grow. It's not S. A process called germination. Germination is the process by which an organism grows from a seed or spore. The term is applied to the sprouting of a seedling from a seed of an angiosperm or gymnosperm. The growth of a sporling from a spore, such as the spores of fungi, ferns, bacteria, and the growth of pollen and the growth of the pollen tube from the pollen grain of a seed plant. So the pollen tube from the pollen grain of a seed plant. Also, that term is also applied. Applied. So you can also call that germination there. So this is a process in which an organism grows from a seed or spore. You can apply that to sprouting of a seedling, or a growth of a sporling, or the growth of the pollen tube from the pollen grain, which is interesting. So anyways, sprouting is a natural process by which seeds or spores germinate. And And yeah, so sprouting or growth of a sporling. This one is sprouting of a seedling. Essentially the same thing, just growing. So sprouting is a natural process by which seeds or spores germinate and put out shoots, and already established plants produce new leaves or buds or other newly developing parts, experience further growth. So basically, it's just growth. So here's a sunflower time-lapse with soil, cross-section showing how the roots and the upper plant of the plant grow or sprout. So it's just, this is just germination. And it's the growth from a seed or pollen tube from a pollen grain. Very interesting. So it's rising upwards like that. Make this a bit smaller there. It's pretty interesting GIF time-lapse showing this sunflower time-lapse with soil. It's pretty remarkable. It's getting bigger and bigger. And then notice the root going down and it's going up. Fascinating stuff. All right, let's continue further. So this one right here, imbibition is the first step in germination. Let's pronounce this one.
Imbibition.
Yeah, so imbibition is the first step in germination, whereby water is absorbed by the seed. And once water is absorbed, the seed undergoes metabolic changes, whereby enzymes are activated and RNA and proteins are synthesized. Once the seed germinates, it obtains carbohydrates, amino acids, and small lipids that serve as building blocks for its development. These monomers are obtained from the hydrolysis or water breaking. Yeah, and here I'm just going to quickly just search it up. So from the hydrolysis, go back up to our earlier note here. Hydrolysis, ancient Greek hydro, water, and lysis to unbind, is any chemical reaction in which a molecule of water breaks one or more chemical bonds. So that's very, very interesting here. All right, so let's continue backwards over here. Search hydrolysis, and we are here. So these monomers are obtained from the hydrolysis of starch, so the breaking of starch and proteins and lipids that are stored in either the cotyledons. Let's see the pronunciation here.
Cotyledons.
Cotyledons or endosperm. Germination is completed once embryonic roots called radical, or let's see this.
Radical have.
Radical or radicali, I'll call it radical, have emerged from the seed coat. Yeah, so at this point, the developing plant is called a seedling, and its growth is regulated by its own photoreceptor proteins and hormones. That's very interesting. And this right here, a cotyledon. Let's see this.
Cotyledon.
Yeah, so a cotyledon, again, this is where monomers obtained from hydrolysis of starch, proteins, and lipids are stored in either the cotyledon or endosperm. So a cotyledon or seed leaf, from Latin cotyledon, is a significant part of the embryo within the seed of a plant and is defined as the embryonic leaf in seed-bearing plants, one or more of which are the first to appear from a germinating seed. So essentially, the first leaves that are there. Species with one cotyledon are called monocotyledonous. See the pronunciation here.
Monocotyledonous.
Or monocotyledonous, monocots. Plants with two embryonic leaves are termed dicotyledonous.
Dicotyledonous.
Or dicotyledonous, or dicot. So there's the dicots we were talking about. So it has two embryonic leaves in this setup here. So here is a setup here. Comparison of a monocot and dicot sprouting, or dicot. Let's see the pronunciation actually.
Monocot and dicot.
Yes, monocot and dicot. So you have here. So the monocot, it looks like it doesn't have any other. It doesn't have a leaf there, but it itself is one, I believe. So the visible part of the monocot plant left is actually the first true leaf produced from the meristem. Yeah. So the visible part right here, that is the first leaf. And you could store some of that monomers inside here. And then produced from the meristem, the cotyledon itself remains within the seed. Yeah, so that's an interesting part here. So this is a monocot. So the visible part of the monocot plant is actually the first true leaf produced by the meristem. So that's the visible part right of the leaf. The cotyledon itself remains within the seed. So that's still inside the seed, which is quite interesting. Yeah, so this part's just a bit confusing what they mean by this. We'll get to a comparison one right here. But then the dicot, you can see there's two embryonic leaves there. So that's an obvious one there. This one right here, this could be viewed as the first true leaf produced from the meristem. But then here, the cotyledon, or let's see this again.
Cotyledon.
Or cotyledon itself remains within the seed. So is it here? But isn't that the first true leaf? It's a bit confusing. This is from Wikipedia. And here's I just took this image from this other source right here. This one is from biologycorner.com. So you can see it right here. This is two cotyledons or seed germinates outwards, and you have the first two the embryonic leaves over there. And then for that's for a dicot and monocot right here. This grows over there, and you just have that one leaf there. Yeah, so you have one leaf there. And then you have it over here, you have two. So this part could be talking about this one right here. This is the first true leaf. It could be talking about this right here, and then the actual cotyledon is still inside. So that could be what they're talking about. Here when they say remains within the seed. So I think that's what it means, and then it finally grows up and it grows up bigger there. Or something like that. So yes. But yes, basically you have two over here, and then this one is just one there, or not two. And call that a monocot dicot. Very, very fascinating. And now that endosperm is that inside part. Let's take a look here. And here's a wheat kernel. Note that a kernel is broadly a synonym for seed, or synonym for seed. So it means the same thing. Essentially. So endosperm is inside here. Remember that was a two nuclei merged with that other pollen grain, or that male gamete or sperm. So it emerges over here, and then there's a brush on top there. Then you have this bran. So the bran is, I think, that's that outer coating there. There's a germ inside there, and a composition by weight, and 3% is the germ. 14% is the bran, and then you have the endosperm is about 83% there. So most of that is in the endosperm for nutrients and so on. It's fascinating stuff. Let's continue further here. And so the endosperm is a tissue produced inside the seeds of most flowering plants following double fertilization. And here, let's do a search right here. Let's go back to our double fertilization when we mentioned it. So we had two sperm nuclei, hence called double fertilization, then passed through the pollen tube. One of them unites with the egg nucleus and produces a zygote. The other sperm unites with a two polar nuclei to produce an endosperm nucleus to produce endosperm. So yeah, so you have this double fertilization. You have two sperm nuclei going in. One goes to the egg, and one goes to this polar nuclei, forms the endosperm. And the other one forms the zygote with the egg. That's very, very interesting. All right, so let's continue back over here. Oops, right here. Let's click here and do search. And we are back here. All right. Yeah, so that's an endosperm. And then bran, also known as Miller's bran, is the hard outer layers of cereal grain. So this is cereal grain or wheat kernel there. The germ of a cereal is the reproductive part that germinates to grow into a plant. It is the embryo of the seed. There's the embryo there. That's the one that grows up. And let's go further. So a cereal is any grass, which is a type of flowering plant, cultivated or grown for the edible components of its grain. Botanically, a type of fruit called a caryopsis.
A caryopsis.
Or caryopsis. Yeah, so the grain is essentially a fruit called caryopsis, composed of the endosperm, germ, and bran. That's very, very interesting. So the seed coat in the mature seed can be a paper-thin layer, e.g. peanut, or something more substantial, e.g. thick and hard in a coconut, or fleshy as in the pomegranate. Yeah, so this is the right here. Yeah, this is that bran, also the outer layers of cereal grain. Grain is just the fruit. Yeah, it's a fruit / grain there, or fruit / seed. The seed coat. Yeah, the seed coat. Yeah, we mentioned it over here actually. Scroll back up here. Remember this part right here. At this point, the developing plant is called a seedling, and its growth is regulated. Shown as before. That's germination is completed once embryonic roots called radicle have emerged from the seed coat. Yeah, so the embryonic roots come out from the seed coat, and the seed coat is Yeah, it's just that so the seed coat in the mature seed can be a paper thin layer, i.e. peanut, or something more substantial, thick and hard in a coconut, or fleshy as in a pomegranate. Just to illustrate it, here the seed coat of a pomegranate, that's fleshy like that, and inside's a seed. And there's a seed coat there, very interesting. And the radicle would be the roots that shoot outwards from the seed outwards past the seed coat. It's very interesting. So unlike animals in which growth is determinate, i.e. ceases when the adult state is reached, plant growth is indeterminate as it is an open-ended process that could potentially be lifelong. Plants grow in two ways, primary and secondary. In primary growth, the shoots and roots are formed and lengthened. So that's a primary one. And then the apical meristem produces the primary plant body, which can be found in all seed plants. During secondary growth, the thickness of the plant increases as the lateral meristem, surrounds the established stem of a plant and grows laterally, produces the secondary plant body, which can be found in woody edicots. See this?
Eudicots.
Or eudicots, which are clade of flowering plants that are dicots, such as trees and shrubs. Monocots do not go through secondary growth. The plant body is generated by a hierarchy of meristems. The apical meristem and the root and shoot systems give rise to primary meristems, the protoderm, ground meristem, and procambium, which in turn give rise to the three tissue systems, dermal, ground, and vascular. And remember, the dermal is the outer skin, and outer coating, ground is everything in between the vascular and the dermal. So MES notes continue further here. So here's the types of growth right here. Let's make this a bit smaller. So you have growth right here. This figure is from cas.miamioh.edu. And this is for digots, have both of these. So monocots do not go through secondary growth. So primary growth, so growth in length. So this is the primary one, everything grows long. And you have the production of branches. Here, this grows long, this goes outwards, this goes up and out. Growth in length, production of leaves, etc. Production, and then production of branches there. And that's primary. And then the secondary growth in girth, in stems, and production of, roots here. Everything grows laterally outward. This grows outwards there. Not this arrow inside. The arrow goes out. So switch that arrow up, and this goes up like that. This goes like that. So it's very, very fascinating stuff. Alright, so now let's continue even further. So now let's look at the types of this meristem right here. So the meristem cells mature into three primary meristems. This is the protoderm, procambian, and ground meristem. Mentioned it over here. So the apical meristems in the root and shoot systems give rise to primary meristems, protoderm, ground meristem, and procambian, which in turn give rise to the three tissue systems over there. That's very interesting. So let's take a look at over here. Let's make this a bit smaller. And let's continue. So the meristem cells mature into three primary meristems. Protoderm gives rise to dermal tissues, or the epidermis, the coating. Procambian gives rise to, let's see the pronunciation actually.
Procambium.
So procambium gives rise to vascular tissues. And the ground meristem gives rise to ground tissue. So we have the apical meristem, the one that goes all the way up, the highest one. And then you have three primary meristems. You have the protoderm is on the outside there. There's the ground one inside, and there's a procambian here for the vascular setup there. And then as they grow, what you have is vascular bundle over there, the xylem/phloem. This figure is from bio.miami.edu. Yes. Continue further. So vascular tissue is a complex conducting tissue formed by one or more, formed of more than one cell type found in vascular plants. I think we're talking about the phloem and xylem. So here is the celery stalk. So cross section of celery stalk showing vascular bundles, which include both phloem and xylem. So it's very interesting. Bundles there, phloem and xylem there, and everything else ground tissue inside, and then the outer coating is a protoderm. The protoderm creates that dermal tissues or the epidermis. Very fascinating. Let's continue further here. This is a setup here. Let's see how these look like here. So that's that vascular bundles that we were talking about. We can make this bigger and we'll zoom in. All right, and scroll back up here. So go to the vascular bundles there. That includes this phloem and xylem. So if you zoom into these, so cross-section view of celery under microscope, 8x. So this is from biology by Napier.com. So Fig 4, drawing of the tissues of the vascular bundles. Or let's go look at Fig 2 first and zoom in. So we're zooming in a lot there. So Fig 2, cross-section view of celery under microscope, 8x. So there. And if you zoom in even further, Fig 3, magnification of the vascular bundle clearly shows 3 distinct layers. The darkest dyed tissue being xylem, 36x. So there's a xylem. That's a flow of water, etc. There's the phloem, the flow of the sugar, etc. And there's the vascular cambrium. Let's go to that vascular cambrium. If I just do a quick search, actually, we didn't actually search this. We didn't actually type this cambrium in here. I'm going to put a note right here. Whoops. All right, here, yeah, so I just quickly just paused video and zoomed out and added these notes here. So the procambium gives rise to vascular tissues. It also produces the vascular cambium and cork cambium, which are secondary meristems. Vascular cambium produces secondary xylem inwards. So going inwards, the xylem is producing that inwards, and secondary phloem outwards, phloem on the outside there, producing in between there. And cork cambium is one of the many layers of bark. So inside the outer layer, you'll have some of the you'll have that lateral slash secondary meristem on the outside, and this one's in between in that that vascular tissue area. So anyways, let's go back and zoom in here. So and there's that vascular cambrium. There's the phloem inwards, and I mean the phloem is outwards, and then the xylem is inwards. So this is if I erase this vascular cambrium inwards, you can see this is the xylem's here, so inwards is towards this way. So it's inwards towards xylem and then outwards in this side to the phloem there. And so this is 36 times magnification, which is quite remarkable. Going further, so Fig 4, drawing of tissues of the vascular bundle. So here's the setup here. So you have that celery stalk, you have the parenchyma, so the other ground tissue, and then you have the parenchyma over here, and there's epidermis, there's a phloem on the outside, there's a vascular cambrium inside, there's the vascular tissue, there's a xylem over there, and there's a parenchyma out there. That's very, very interesting setup here. And we can zoom out. Now look at this figure right here. Yeah, so this figure right here, this is from, let's make this a bit smaller so we include that there. This is from, this figure is from Holganics, Holganics or Holganics.com. So you have, this is the cross-section here. Cut this over, move it over there, and look at it from the top view. So you have the xylem inside, then you have the phloem over here on the outside, and that's a vascular tissue, and then there's a ground tissue in between, and there's a dermal tissue, the epidermis. And remember again, the ground tissue of plants includes all tissues that are neither dermal or vascular. Very fascinating.